So there is a girl whose dad in India, right? And of course, I had no idea where they were talking about in India, right? So I looked it up, right? And it turns out not just this, but over here is also India, as you can see on the gray part. And this is the part of the country in which they live, right? And so near Bangladesh, right? And poor Nepal as of late. So what pathogens cause meningitis, right? What could cause meningitis? First of all, what is meningitis? Meningitis is an inflammation of the meninges, which is the membranes that surround your brain and spinal cord, right? Whenever they say itis, is typically they're referring to as inflammation, right? So whenever anything ends in itis, they're really talking about inflammation. So in this case, what's causing it? What type of organism in this case study? No, it's bacterial. It's bacterial, right? So Streptococcus pneumoniae, Haemophilus influenzae, Neisseria meningitis, which is the causative agent in this case, and Listeria, right, which has been in the news lately, right, and is the bonus one that I'm going to give you guys, can all cause infections that can lead to meningitis, okay? So viruses somebody mentioned, and there are numerous viruses, several enteroviruses, ones that infect your gut, the mumps virus, and one of my students this morning was like, you know, I've had my titer checked. I know a lot of people are getting ready to go into nursing school and stuff like that, and you have to make sure you're up on your immunizations. And he said, you know, I've had the measles, mumps, and rubella shot, but yet my rubella titers are good. I've got antibody levels, and so are my measles levels, but my mumps was 0. And the problem with this is that it's not a very effective vaccine for mumps. The other two, yes, but not so much with mumps. So we still see, and we've had outbreaks of mumps and issues with mumps because it unfortunately isn't the best vaccine, but because of it though, we do see less than probably if we didn't have a vaccine against mumps. The arthropod-borne viruses, especially in this area, West Nile is of concern, right, that it could lead to even infection of the brain itself. And St. Louis virus. These are ones transmitted by mosquitoes. And so these are the ones too, like in Louisiana, they trap mosquitoes specifically and test them for West Nile. So if you live in an area where they spray for mosquitoes and you see the truck a little more often than you normally do, it's probably because they've gotten a positive result in their trap. But like I said to my other class, you know, not a reason to, you know, hibernate and never go out. We have repellent, right? Protect yourself, and obviously stay away from areas that are real problem spots. So in most of the cases, you know, you just have a mild illness, but there is sometimes where it can be neuroinvasive. Fungi is another one, and I didn't list any fungi here. But not too long ago, they had a medication that was contaminated with aspergillus, with a fungus. I want to say it was like a steroid injection or something like that, and several people got meningitis from that. And so that it can happen. It's not as common, and obviously that was a problem with the manufacturing, unfortunately, of that drug that led to that higher incidence. Parasites. Right, there are several parasites that also can cause problems. But our two big offenders usually are bacteria and viruses. And we'll talk about some of the ways in which we distinguish between the two when they do the testing. So which pathogen is most often associated with epidemics and the meningitis bout is Neisseria meningitis, the one that's commonly referred to as meningococcal. And the reasoning for that you'll see in a moment when we look at the actual organism. The meningitis belt is actually a long, thin region in Africa, right? This case is dealing with an outbreak in India, but they commonly happen in Africa along this stretch. So the organism is a gram-negative bacteria. So it would be pink, right, when we stain it. It's a diplococcal, which means that it tends to stay in pairs like you see here. And in this picture, you can see a white blood cell, probably a macrophage or a monocyte, engulfing lots of the bacteria. And when they run the tests, right, they'll do a stain and look for the bacteria as well as these white blood cells. You'll see an increase, of course, when you have infection with the white blood cells. So what type of sample do you think they're going to take? There's cerebral spinal fluid, right? So your CSF. And they're going to do what's called a lumbar puncture to obtain that. And that there's a portion in the lumbar region, so they're in your lower region, where your spinal cord itself tapers. So there's less likely when they go in with the needle that they're going to damage the spinal cord and hopefully just get a fluid sample. And then, as I said, they're going to stain it, they're going to gram stain, and they're going to look for the bacteria. The other test that they'll run on this is they'll do a glucose test to determine the level of glucose. Glucose significantly drops in your cerebral spinal fluid when you have a bacterial infection, because the bacteria, of course, are eating the sugar that you normally would be eating. With a viral infection, of course, we're not going to see viruses under the microscope, right? If it's bacterial, we should be able to see them under the microscope. You're going to have a different response of white blood cells. It's not going to be as aggressive as you see with bacteria. And then, of course, you're also not going to see the drop in glucose because viruses don't eat glucose, right? They infect our cells. So that's one of the ways they can make that distinction between bacterial and viral. And of course, especially with bacterial, we want to know the source of the infection so that we can effectively treat. So some other information they give you in the case study is how many people are sick. So there were 2,500 people that came down ill and 230 deaths. That's pretty significant in the past month. And it's within a population of about 140,000 people. So we can figure out what's called an incidence rate. And epidemiologists that study outbreaks and the transmission of disease do this all the time. And what incidence is the number of cases of a disease per a specific population occurring over a given time period. So we have enough of the numbers here to be able to calculate that number. And it's typically calculated and then multiplied by a factor of 10 to help in understanding the numbers. So to give you an example of how it's typically represented, if we just do the division straight up there, we end up with 0.01. What does that mean, right? We could probably put it in a percentage. That would be, 1.8%. That might mean something. But typically the way the epidemiologist will present the material is, especially diseases that have a really high incidence, it's usually in the hundreds of thousands, especially when we're talking about the entire United States. But for this case, if you multiply by 1,000, we end up with a whole number that we could understand. And the way we would say that is that 18 people per 1,000, right, became ill from this infection. But then it doesn't sound quite so bad until you get to the next rate, when you factor in how many people have what's mortality, how many have died, right? And the way this is calculated is the number of deaths of the disease over the time period by the number of people with the disease. So in this case, it'd be 230 divided by 2,500. Again, we get this weird decimal. If we put it into a whole number by a multiple of 10, it would be 92 people per 1,000, or 9%. That's pretty significant, especially if you happen to be in that 9%. So the next morning, and it's her father that's ill, right? He was vomiting, he's very sensitive to light, he has a stiff neck, and my little pop-up didn't work. But stiff neck is one of the key indicators of the possibility of meningitis. And the reason why a person ends up with a stiff neck is this inflammation, right, surrounding the spinal cord and brain. Those membranes called the meninges are inflamed. When they become inflamed, everything is cased in bone, right? Your skull, your spinal cord. So there's really no flex, no way of moving. So the individuals really do not want to move their neck. It becomes stiff from them trying to hold it in a particular position. You ask them to move it, they're in excessive pain. So if you walk into the ER and you complain of a stiff neck, they're probably going to take you right in and do a lumbar puncture, right? After they ask you if you've been exposed to anybody with meningitis that you're aware of. So that's one of the key indicators. And then, of course, this particular rash that we'll talk about in a moment is pretty specific to this type of infection. So first of all, how is it spread person to person? How's it spread? Now, it doesn't start out at your brain or spinal cord, right? And for this particular pathogen, it's respiratorily transmitted, right? So you need to have direct contact or indirect contact with things that have been contaminated by someone's respiratory droplets. And again, because it initially will infect the respiratory pathogens, so that's where it's multiplying, that's where it can be spread, even when it has spread to the meninges. So what's bad news? What characteristics enable it to cause the damage they do? So this guy has got quite a few tricks up its sleeve. The pathogen produces a protease, which destroys the antibody found in mucus. And which antibody is a good review for our test coming up? Which class of antibody is found in our mucus? IgA. Good. Y'all been studying. It also produces an anti-phagocytic capsule, right? So this capsule helps them evade phagocytosis. And so therefore, they're able to grow to large numbers, enter into the bloodstream, and that's the most common way your central nervous system is infected, is when you have large numbers of whether it be bacteria or a virus circulating in your blood, that it crosses the blood-brain barrier, right, into that usually relatively restricted area as it goes to infection. So it is, as we said, already a gram-negative bacteria. The bad news with that is upon destruction by our white blood cells, it releases endotoxin. This is the lipid A component of the lipopolysaccharide that makes up the outer membrane of gram-negative bacteria. So as we're trying to destroy these guys, we just make the situation worse. This particular endotoxin triggers our release of tumor necrosis factor, right? So we're not releasing it, right? They're causing us to release it. And what this does is it damages the capillaries and causes the bleeding, and it can even cause shock if it's present in large quantities in the body, right? So people that can go into, so sometimes when you have really serious infections, right, and people, by the time they get to the doctor, there really isn't much they can do for them. It's when it's reached these really extreme levels, right? And sometimes they refer to it as being septic, right? And this is when infection or toxin have gone system-wide through the bloodstream. So a friend of mine who works here, he had a pretty serious infection once, and he said, I just really didn't even fathom how close to death I was, and how grateful I am now. And he's like, I kind of can understand about what you teach now. He's a business teacher, so he's like, that stuff is kind of important, just a little bit. So that particular rash, sometimes referred to as petechia, happens when the blood vessels in the skin are being damaged by the release of this particular toxin as we're killing the bacteria. So one of those kind of catch-22s. So in this case, she's not able to get to a hospital, but they're giving out antibiotics. They're giving out chloramphenicol. Although there's a range of antibiotics that are effective, like several of the penicillins that are, because remember it's gram-negative, so straight up penicillin's not going to work, but you need the broad spectrum penicillins like ampicillin. Under epidemic conditions in Africa, typically they prescribe chloramphenicol, and the reason for that is it's very inexpensive, and it's long-acting, and it is effective. This works differently than how penicillins work, and we'll talk about that in a moment. It is normally not used as a first line of therapy, though, here in the United States, because there's a rare complication of aplastic anemia that can happen, right? So it's not our first choice when it comes to infection. The way chloramphenicol works is it blocks the peptidyl transferase action. So this is the enzymatic action of the ribosome. This is when it's putting together the amino acids to form the proteins. So, and it specifically affects the 70S prokaryotic ribosomes, right? So it affects just the bacteria, and it doesn't affect our ribosomes. So that's what we mean by mechanism of action. Remember, the penicillins interfere with the bacteria's ability to make what? Very important component that only their cell walls have. Bacteria. Peptidoglycan, right? Interferes with peptidoglycan. So this is a different target. So how could the family help prevent further spread of the pathogen to others in the tribe? Number one thing, right? Proper hygiene, right? Covering the mouth when coughing and sneezing and frequent hand washing, right, are number one. In addition, right, you want to, if you're in that environment, you could have potentially touched something, you don't want to expose yourself, right? So you want to avoid touching your eyes, your nose, your mouth, right, because that increases the risk. Whenever we say that, it seems like we can't help but want to touch our face, right, when we say don't do it. And sometimes they can give prophylactic antibiotics, like rifampin. So they'll give it to people, and this will also help with people that are asymptomatic carriers during an outbreak, where they're not showing symptoms, but they have it growing their respiratory passages in there and transmitting it to others. So that's it for meningitis. I don't think I made any changes. I had already posted the changes I had made to this one for you guys, right? It's the other ones I didn't. Okay, so next up is it hepatitis, or it is going to be hepatitis? Why not? These last couple give me stomach aches. I didn't realize how many, like, stomach ones I had picked. But I was trying to pick, different ones, bacteria, viruses, that kind of thing. So this poor lady, right, unfortunately went out to eat. Same thing that happened to me last week. I chose the wrong place to go. And I can feel her pain. Although thankfully I did not get this particular infection, probably some other bacterial infection. So she's, one of the symptoms here that they have highlighted is jaundice, right? And it's an interesting symptom or sign, you could say, because you can actually see this in individuals. So it's when you have yellowish discoloration to the skin and to the eyes, and this particular pigment that caused this yellowing is called bilirubin. And nope. This is a brownish yellow substance. It's a byproduct of the breakdown of heme, which is the component of hemoglobin in your red blood cells. So that's actually where it originates from. But it is processed, removed from your blood, right, by your liver, right? And that's one of the important points here. And then it's excreted into your bile. And bile is used to help emulsify your fats and help in digestion, right? So it goes into your digestive tract. And that's one of the main reasons why your feces is actually brown color, is because of this particular pigment, this yellowish pigment. And it's actually why bile, to a certain extent, is green. The yellow component of the green is from the bilirubin. So this individual is severe jaundice in this picture, because you can see the whites of his eyes are yellow. I've actually seen this in real life. It's really frightening because, it's yellow. And the individual that I saw in a doctor's office was, his liver was failing. He had had a liver transplant and it was failing. He walked in and right away I was like, what the heck is wrong with this person? I said to the nurse, I was like, what is wrong? And she told me, that he was in liver failure. So it's one of the indications, right, that there's something may be wrong with your liver. And notice I say may. Okay, so what type of diseases? So if she's jaundice, what could be going on, right? Number one, this case study ends up being what? hepatitis, right? But there are several things that can, and that just means an inflammation of the liver. But there are several viruses as well as a viceroid that can cause the damage to the liver, and therefore end up having the buildup of the bilirubin, and you can become jaundice. So hepatitis viruses A, B, C, E, and G. D, we have found out, is actually a viceroid. So a little review from the beginning of the semester. What is a viceroid? It infects a virus, and typically it's inside the hepatitis B virus. But what is it made out of? So viruses are DNA or RNA surrounded by a protein coat, right? What's a viceroid, though? It's only one of those. It's RNA. It's just RNA. And it needs a virus to be able to, travel from one place to another. So if you have hepatitis B, you may also have hepatitis D, because that's the virus that it infects. The viceroid infects. There are two other major infections that thankfully another way we can rule them out is that we happen to live in the United States, and they are not common in the United States. And that would be malaria, which is caused by a plasmodium, which is a protozoan. And there are several different species of this genus of protozoans that cause infection, lots of plasmodiums. So it depends on the organism to how bad the infection will be to, depending on the life cycle that we'll look at briefly in a moment. Can anyone think of another one? It actually kind of gets its name from the fact that people tend to look yellow. Yellow fever. So yellow fever is caused by a virus. And the good news for us, although these are both transmitted by mosquitoes, and we certainly have mosquitoes here, we do not have the wild animal populations harboring these pathogens. So they don't have the protozoa, we don't have the virus here. At one time we did actually have malaria in the United States, but it has been eradicated, thank goodness. And I don't think they've ever had yellow fever. I can't remember if it was the Panama Canal when they were doing that, if it was just malaria or if they had yellow fever outbreaks too. I can't remember. I should know my history better. So for malaria, this is kind of a twofold punch when it comes to malaria, because this one will infect the red blood cells and damage the red blood cells. That's part of the cycle, and they reproduce inside the red blood cells. And the other part of the cycle is that they reproduce inside the liver cells. So not only do we have destruction of the red blood cells, right, which would increase the level of this pigment, the organ that's supposed to filter out this pigment, your liver, is also being damaged by the infection. So all our hepatitis, and notice the mode of transmission. Fecal oral is the most common for hepatitis A. And you'll see, and we'll talk about in a little bit, the vaccine that they have for this. Before the vaccine, it wasn't uncommon for individuals to get this type of infection. Hepatitis B, and the other one on this list is E, and I fixed this morning. It said Tide or something instead of United. My students caught it this morning. Hepatitis E is not common in the United States, right? So although that one's also fecal oral transmitted, one of the ways we can rule that out is that it's very rare in the United States, right? So other parts of the world where you see that. So notice, as I said, hepatitis A is a virus. It has an RNA genome, where hepatitis B is a DNA virus. Hepatitis C is an RNA virus. But notice too, with these guys, as well as G, and then of course D, remember, is a viceroid that typically infects the hepatitis B virus. So you don't only have this if you have hepatitis B, you might also have this viceroid, which of course is just RNA. So because it infects the hepatitis B virus, that hepatitis B vaccine helps protect you against hepatitis D, right? Because hepatitis D is inside of B. But, very restricted on exposure, right? Blood, needles, body secretions. B, for mothers, it can be transmitted via the placenta and sexually, right? So, you know, the likelihood of coming in contact or doing any of these things is pretty slim, right, for most individuals. So again, jaundice indicates potential damage to what organ? The liver. And so the thing with yellow fever, though, it's destruction of red blood cells and the liver not being able to keep up. And so that's another thing for babies. A lot of times babies are born and they're a little jaundice, and depending on how quickly their liver comes online. And that's what, it's just that, you know, their liver just isn't up to snuff yet, you could say, right? And over time, hopefully, it's able to do that. UV therapy, UV light actually will help break down the pigment, and that's one of the things they'll say, just take your baby outside in the sunshine for a few minutes. Yeah, and sometimes in severe cases where it's really not breaking up, they'll put them in the incubator with UV lights, yeah, to help with that. So for this particular case at the doctor's office, he starts asking her all these questions. What is he trying to do when he asks her, have you traveled overseas? Why does he? He wants to rule out the possibilities, right? So we've talked about hepatitis is a possibility, malaria is a possibility, so is yellow fever, but two of those we can knock off if she hasn't traveled overseas, right? And she hasn't, right? She has not traveled overseas, so we can eliminate those as well as hepatitis E, right? Because that one I said is not common in the United States, so she would have probably had to travel. IV drug use. Again, he's trying to limit it down, right? As to what type of hepatitis it may be. Is this a blood-borne type thing? I'm sure most people don't relish having to ask their patients this question, but it's an important question and to answer honestly. I wonder how many actually do. Had she eaten at a restaurant that had been linked to numerous cases of hepatitis? And unfortunately for her, the answer was yes. So in this case, right, whenever stuff like that happens, it gets reported to the state health board and it gets disseminated to doctors' offices, right? So he was aware of this particular outbreak, and so he was able to add this to his list of questions. That's what made him do that. So what test is used for the presence of hepatitis virus? They're going to do a blood sample, and in this case they're actually going to test to see for antibodies that she's produced against this virus. And this particular test that they commonly do is called an indirect ELISA, and those of you guys in microbiology lab this week will be doing that, and I just set it up this morning. It's probably why you couldn't find me. I was in the lab putting little antibodies in tubes for you guys. So they use actually antibodies, and actually let me put it up there. ELISA stands for enzyme-linked immunosorbate assay. So they actually use antibodies to detect the presence of other antibodies, except the antibody that they're using for the detection has an enzyme linked to it. And when you add a substrate, a color change, sorbate, that's what refers to a color change. In assay, you have to do it stepwise. So you have to do the steps in order, otherwise you won't get accurate results. This is a very common type of testing used very widely in research nowadays. Although this one is antibody-based, there are others that are antigen-based. Pregnancy tests that you buy over the counter, right, are this technology using antibodies. The rapid strep test they do in doctors' offices. It's an indirect ELISA, I believe, for heartworm test in veterinarian offices. Right, they're looking for the antibodies. Those worms are too big; they're not going to be in the blood, right?
You can do a direct smear, look for the babies.
Oh, okay.
You can do an antigen test.
For the you do the ELISA test for the antibodies on the blood. It's a little snap kit.
Yeah.
Yep. So. And then you can do direct testing, detection of nucleic acids, and this is through polymerase chain reaction. It's a little bit more expensive testing that's sometimes performed depending on the situation. So back to our chart, right? So for her it happened to be hepatitis A, oops. And what was the reason for that? Well, she ate at that restaurant, right? And they knew it was a hepatitis A outbreak. And the problem here is you can get this from eating something that's been contaminated. So notice the transmission is fecal-oral, which means something was contaminated with fecal matter containing this virus, and she ate it, right? Do you guys remember what it was for this one? Green onions in the salsa, right? So what human byproduct is the virus found in? Feces, right? So, what are all the possible ways the green onions could have been contaminated? The crop itself could have been contaminated with contaminated water. My stomach is still gurgling. Okay. Contaminated soil from fertilizers used. The workers, right? When harvesting, when packing, when transporting, or last place at the restaurant. It could have been anybody in that chain, right, that led to the contamination of this food product. So, as you can see, right, they had what was it I wanted to say? So this is when they ate, when the individuals ate, right? So apparently this was not the best day to have eaten there. And then you can see, you know, a good about, what? So if we say 21 to onset, right? The largest was about 20 days later, like 2 weeks later, right? Significant amount of time before people were really showing illness. Can antibiotics used to treat hepatitis caused by a virus? No. What do we take antibiotics against? Bacteria. So we're not sending her a pill on her way. She's not too happy about that, I'm sure, because there are any antivirals we can give her. Currently, no. She has to wait for her immune system to take care of it. And you can be quite sick from this. Before widespread vaccination like we have now, I remember as a little kid, my parents, you know, going out to dinner one night, and then a week or so later, grandma coming to take care of us, because my parents could not. They were so ill. So you can be significantly ill, right? So a few weeks or even months, right, depending on the person's immune system, how well they'll recuperate. So there are two vaccines, the hepatitis A and B vaccines. We do not have one against hepatitis C currently. But again, with hepatitis C and B, you can limit your possible exposure to these things. And so where hepatitis A, not so much, right? So hepatitis A, you're suggested to receive 2 doses of the vaccine is significant, and they need to be given at least 6 months apart. And they start vaccinating as young as 1 year old, right, of age, so 12 months of age. And since this, you know, we've seen significant decreases in hepatitis A. As well as hepatitis B, the vaccine, we started routinely giving it to children in 1991. And in fact, I think this is one of the ones that most places they give at birth even, right? So your child is given this vaccine before they even leave the hospital. If you donate blood, if you got your Hep B vaccine a week ago, should you donate blood? No. And why not? You're going to have the vaccine in your blood, right? It's a weakened form of the virus. They test for this, of course, in blood samples because they don't want to transmit it to someone else. They don't know unless you tell them, and if you tell them, they're not going to take your blood because they can't distinguish between the vaccine and the actual infection. And so they don't want someone to get the infection from your donated blood. Right? So if it tests positive, they're going to toss it, right? They're also going to inform you, okay? They'll run additional tests and they will inform you. So you're cautioned, you know, if you're of age and whatnot, one of the things they should tell you when they give you this vaccine is not to donate blood. And I forget what the time frame is. But I gave you guys the fact sheets that I wanted you guys to read for these, and this is why I asked you questions so you would actually read them. And this is right off the CDC's website. This is exactly what they would give you when you get vaccinated. Very few people actually read these sheets. So you might be among the few, right, who have actually read them now and are informed. Do you expect her to return back to work in several days? No, she's probably going to be out of work for weeks, right? She's looking at a significant recovery period, unfortunately. How would you prevent this type of future outbreak? Well, of course, the list, the same from before, right? We want to make sure that the water's not contaminated, the fertilizers, and the workers, right, which is 3 through 6, we're going to teach them proper hand washing and proper hygiene. And one of my students in my last class brought up the fact that there have been some issues in the past with people defecating, right, when harvesting in the fields, right, because they don't have any place to go to the bathroom. And so educating people about the possible hazards of doing something like that. And you would think that things like that don't happen, but in fact they do. So, kind of scary stuff. So then I continued on the good old trail, and we did Shigellosis. So Shigellosis, this is another bacteria, and with anything else, right, it's very important that we maintain hand washing. So there was an outbreak in a preschool in this particular area, and so they were sending home all these pamphlets to try and get parents to help reinforce at home. So what types of pathogens could be inhibited? And basically, I just flipped the question a little bit and said to you guys, what could make us sick, right? What could be found on your hands that would make you sick? Bacteria, clearly, right? We're aware of that, right? But what else? Viruses, right? The same list from before. Fungi, right? And parasites. Of course, the top two are the two main offenders, right? Bacteria and viruses. Can you wash these off with hand washing? Absolutely. Absolutely.
Is that like related to what's going on with the red dots on your hands? You never heard of that?
No.
That's going on. Like, it's common in kids, but like adults can get it too. With the red dots on your hands and legs.
I do not know about that. I'll have to do some investigating.
Because one of my friends got it from her friend baby.
Yeah, but some type of infectious
Yeah,
infection.
something like it has something
I don't know what it is,
to do
though. Don't know.
with what's going on.
Got me on that. Red dots is not enough information.
I don't know what it's called, but you, like if you put it in Google, it's just gonna pop up.
I'll have to take a look in my spare time. So, how can pathogens get into our bodies from dirty hands? So the example I give is, obviously, if you have a wound, right, and you touch it with dirty hands, you're going to introduce it into your body, right? What else should you not touch with your hands? Which, of course, the minute we say it, we feel we need to. Your face, right? What on your face? Your eyes, your nose, your mouth, right? These are major entry points for pathogens, right? Don't ever rub your eyes without washing your hands, right? Especially if you've been in the kitchen. Cutting up something, then you introduce it. So we don't want to touch our eyes, our nose, our mouth. Those are entry points. What is one of the most common ways hands are contaminated with microbes from our bodies? Not properly cleaning after defecation. Right? That's the most common way, right? That good old fecal-oral route of transmission. So, right straight from the CDC's website, and a student of mine in the past had given me a video link. I have to admit, I haven't looked at this in a long time. I'm not sure how entertaining it is, but I left it up there. Most people do not follow proper protocol for washing their hands. The major problem is, most people wet their hands, apply soap. That usually isn't an issue. The problem is that you're not scrubbing outside of the water, right, your hands long enough, and then rinsing, right? That's the most common cause, right, of not getting enough stuff off your hands. This one goes so far as to say, the backs of your hands and between your fingers and under your fingernails, right? Which is true, and most people don't think about that. And as I said, the time frame is significant, right? And there's been lots of studies that have been done, right, on the different time frames, and the most widely accepted is about 20 seconds. And so one of the tips they give is sing happy birthday twice in your head, right, to keep track. Most people know that song. Little kids love that song, right? So they love their birthdays. And then run your hands under water, dry your hands with a clean towel or air dry them. So the other issue too is a lot of times, although a lot of public sinks and such that have the knobs that come out now instead of the ones you have to twist with your whole hand, and even some of them are automatic, although there's been some issues with those harboring bacteria within that mechanism. So some hospitals who had them have taken them out since then because of that potential. Especially in a hospital setting, you don't want your water contaminated for sure. But using your elbows, right, instead of your hands to actually close off the faucet. So depending on where you work, I can tell you from working in many different fields, probably the best one was working with disabled children and stuff like that, where you have to change their diapers and stuff like that. And in daycares, they're real particular. I mean, I kid you not, part of my training was someone had to watch me wash my hands. And make sure I followed all the proper steps, and I had to sign a piece of paper that I would continue to do such. So in the one of the restrooms here on campus, the faculty members, we had a little knob to bring down the paper towels. So what we'd always do for the courtesy for the next person is we'd always leave a towel ready, right? So that you would wash your hands, right? You'd go grab the towel, dry your hands, turn off the sink, right? And you'd put the next towel for the next person actually once you took the towel, right?
That's what I do at work.
Yeah. So we had a good little system. Now we have one where we don't have to do that, so we just grab it with our hands. Little precautions like that, especially when you're using public restrooms. Like my son will come into the restroom with me, and because I'm not gonna leave him outside, right? And he touches things, and I'm like, you gotta wash your hands. And he's like, I didn't go potty. I said, you touched this room. You're going to wash your hands before we leave out of here. I don't know what you've been touching when I haven't been watching you. So, proper hand washing is very important. And then what's become very popular too is the alcohol-based sanitizers. And the CDC recommends that you wait until your hands are dry, right? Some people actually rub their hands. You want to actually rub your hands together until they are dry, right? You don't want to just wipe it off on something. So the other thing with that's of concern is that if little children use it too often, alcohol absorbs through the skin, and they can get alcohol poisoning. So one of my students was like, they want them to use it at daycare, and I'm really worried about that. I'm like, once or twice a day is fine. Every hour, no. Right? That's way too much, right? That could potentially lead to exposure. So for Shigellosis, it's a genus of bacteria that can cause this infection. There are 4 different known species. So Shigella flexneri and Shigellosis, which these guys cause about 90% of the cases that clinicians see. A little bit more serious one that can be involved in serious outbreaks is Shigella dysentery. And Shigella boydii is relatively scattered, not very common cause of Shigella. So what's the bad news with these guys? So you're going to have usually acute bloody diarrhea, and this will deal with the organism itself when we talk about it. Crampy abdominal pain, I always mess this one up, tenesmus. Tenesmus. This is a tongue twister. And what that means is this constant feeling of needing to empty your bowels even after they're empty, right? So you've gone to the bathroom and yet you just feel like you just got to keep going. Fever and occasionally vomiting are key features. So why are children in daycare centers at higher risk than others from acquiring respiratory as well as diarrheal diseases? bad hygiene, right? Why are they like making up songs and trying to teach these kids how to wash their hands properly, right? I don't even know how many years it took me to teach my son to sneeze into his elbow, okay? And adults I still see sneezing on their hands, and I'm like, okay, go wash your hands, because you're going to touch something that someone else is going to touch. So the mom is reading on the report, right? The 42 different daycare centers, right? This is a serious epidemic they're dealing with. 600 confirmed cases, 74% of the cases are in children less than 10 years old, right? So we're having a problem probably with diapering issues, right? And people not properly washing their hands. Large concern, and then it goes on to say that 89% of the pathogens characterized were resistant to ampicillin and bacitracin, right? But they were sensitive to ciprofloxacin, what we commonly refer to as just Cipro. So a number of confirmed cases in that particular county, right? And you can see they had a significant spike in the July month, which I think is that, did they say when it is for this one? I don't remember. Doesn't matter. Okay, so how's it treated? Well, mostly mild cases don't require antibiotics, right? We just want to keep the individuals hydrated. However, antibiotics are recommended in severe cases, especially for individuals who have chronic illness, for the elderly, as well as children, malnourished children. HIV patients, remember their immune system is compromised, so this is going to be more difficult for them to deal with. Food handlers, why do we want to treat them? Because we don't want them transmitting it to others, right? Same thing with healthcare workers, we don't want them transmitting to others. And children in daycare centers, because again, we don't want them transmitting it to others, right? Especially them, because hygiene is something they're still learning. So it would appear that Shigellosis is somewhat an opportunistic pathogen. And so this comes into play, remember, when the opportunity arises. So think about our gut microbes, right? So antibiotic therapy decreases the duration of fever and diarrhea by about 2 days and shortens the period that the pathogens actually shed, and therefore decreases the risk of spreading it to others. Oh, virulence. What is so nasty about this bug? So it produces a toxin that is named after it, the Shiga toxin, and this is an exotoxin, so it's actually secreting it outwards, right? So it's growing in your body, it's secreting this. And what it does is it inhibits protein synthesis in eukaryotes by inactivating our ribosomal RNA, okay? So those ribosomal RNAs, right, cannot function. It kills the glomerular epithelial cells, and it can even affect central nervous system, microvascular endothelial cells. Whew, got through that one. It can also lead to hemolytic uric syndrome, which can be quite serious, affects the kidneys, and even seizures. So again, in certain cases, it's definitely important to treat. And then remember, this is a gram-negative bacteria, just like in the meningitis case study. So this one also releases an enterotoxin. Wait a minute, I'm getting ahead of myself. This one is another toxin unrelated to the endotoxin lipid A. Enterotoxin, referring to affecting the intestines. This one causes our intestines to secrete ions, which causes fluid to come back out instead of be absorbed, right? And this is one of the main contributors to the diarrhea. It has proteins that promote its invasion into our cells, and these have actually been linked to particular genes that the bacteria carry. And then the proteins that enable its spread laterally from cell to cell, and this is probably the most interesting, although not very cool. So our M cells that normally antigen sample in our intestines will pass them to the macrophages. The bad news is they can escape. And then also once inside of our cells, these aren't flagella. It can attach to our actin filaments, part of the cytoskeleton in our cells, and use that proteins, those proteins to propel themselves from one cell to another, almost like slingshot themselves through our cells. So of course this can cause cell destruction and inflammation and neutrophils coming into the area. So you're getting destruction of our intestines because of this organism. So I mean, multiple faceted evil, you could say, for this organism. It's amazing that we eventually are able to take care of it on our own. But why is antibiotic sensitivity profile of this pathogen important? So we mentioned that there was some resistance. So number one, do you want to give an antibiotic that's not going to kill the bacteria? No, right? So you want to give one that's going to be effective against the organism you want to give. Plus also think about this. When we give antibiotics, does it just target the bad guys? No. The normal flora get affected as well. And remember we said that this tends to be more of an opportunistic pathogen. So if you're killing off your normal flora and not it, you're just making it a better environment for that organism to grow, right? So we really want to make sure that we target them, right? So what's killed that could help protect us? And that is our normal flora, right? Especially if we're giving a non-effective antibiotic. So I ask you what antimicrobial agents are found in Bactrim. So it consists of two antimicrobial agents. I'm not even going to bother to try and butcher these chemical names. They work differently, but they work in different but similar ways. That seems like an oxymoron. Okay. They inhibit folic acid synthesis. Bacteria have the ability to make folic acid. We do not have this ability. We don't have these pathways. So the way we attain folic acid is from our diet or from the microbes in our gut that are producing the folic acid. So these antibiotics will affect this folic acid pathway. One of them do they not tell us? Oh, it just tells us different stages. So folic acid is important for protein and DNA synthesis, and then as I said, we have it, we need it but we have to acquire it from our diet or our microbes living inside of us. So how do you test for the presence of this pathogen? And so I gave you guys a little bit of hint. They would do a culture, and this is one of the media that we talked about in this course, so it's a good review. Eosin methylene blue. So again, it grew on this agar, so what type of bacteria is it? It would grow. It's gram-negative, right? Gram-negative grow on eosin methylene blue. But you don't see any color change here, right? Most of the pathogenic ones, the really bad ones, actually don't ferment lactose, right? So remember, lactose is one of the sugars that's commonly present, and different formulas also is sucrose. So E. coli, which is a known fecal coliform, although we freak out about it, usually this isn't the one that makes you sick because it's part of your normal flora. It's when you have pathogenic strains, right? Ones that have virulence factors that can do bad things, have particular toxins that make us sick. This one will grow. Remember that metallic green sheen indicates that acids were produced from the fermentation of the sugars, mostly lactose for E. coli. So in addition, serological tests are done to determine which species. And the serological is they use antibodies, again, to determine which species you're dealing with. And so each species has actually been given a letter designation. So dysentery is A, Flexneri is B, Boydii is C, and Sony is type D, serotype D. And again, this is related to that lipopolysaccharide, that outer membrane that they have. Those sugars associated with the lipopolysaccharide are unique to the different species of Shigella. And so we have antibodies specific to those specific antigens, and we can determine which group it is. So, and I think in this case it was Sony, I want to say. So what's the rationale behind this type of public health legislation? So in this it says that the children, right, have to complete antibiotic therapy in two consecutive negative tests for Shigella. These are stool sample checks before they can return to daycare. Why such strong regulation? Yeah, we don't want others to get sick, right? Poor hygiene of a small child, right? You can't always be on top of, right? So we really need for them to not have the infection before returning back. So what activities could you put into place, right? And a lot of it, right, hand washing, and among the children, properly supervising them and informing them. Everyone who changes a child's diaper, which should be sure that the diaper is disposed of properly, right? And again, in this case, if they've got it, they're not going to let them back until they don't have it anymore. And I'll tell you that most of the daycare centers are really serious about this because they understand the risk of infection. So much so that when my son was in daycare, he had his own little box of wipies, right, with his name on it. So, you know, you know, you can get really serious illnesses from, you know, having to change diapers and multiple diapers and not doing it cleanly. So, and even to protect themselves, they wore gloves, right? And some daycares make you even pay for those. I don't mind paying for stuff like that, though. It's worth it to protect everyone involved. So then I went ahead and ruined Strawberry Festival for us. Because Giardia, which is the reason why I picked this one, was because it was a protozoan, right? Something different. But unfortunately, again, this is from somebody eating something. They got this infection. So they had went to a festival and ingested some contaminated food. At first, I think they thought it was just stomach upset, and they suggested probiotics, right, which is in multiple forms, and most yogurts, y'all, have been probiotic forever, right? They're just souping them up and putting specific microbes that have been scientifically proven to help with the gut microbe environment when it's been disrupted. But yogurt has always been beneficial, as long as it hasn't been pasteurized and they kill off the cultures. It has actually live cultures of the bacteria in it. So, oh yeah, this poor little one. He's more susceptible because he's HIV positive. Not a little one, he's 38 years old. So the microbe responsible for this condition is Giardia instantalis. Sometimes you see labia, but I think they've determined that it is actually only one species, and I think they're sticking with the instantalis. It was discovered back in 1681 by good old Anthony von Leeuwenhoek. Remember him? The inquisitive little guy that he is. He, guess how what he looked at to see this? He looked at his own poop. He was a really curious individual. It's a single-cell protozoan. It has two nuclei, so it almost looks like it has eyes. It has multiple flagella, 8 of them in pairs. It has a sucking disc, and this is what allows it to attach to your small intestines. And in the trophocyte form, it's feeding. It does have an inactive form, which is the cyst form. And the bad news with that one is that's the infectious state. I love this picture. So as I said, they kind of look like they have eyes, but those are just the nuclei. This is the trophocyte, and here's that sucking disc I talked about they use to attach to us. They even make cute little stuffed animals of these guys. That's what this is. And then this is in the cyst, and you'll notice here it looks like almost like 4 eyes, 4 nuclei. There are actually 2 of them in here, right, that will bust out of this cyst. So, in the intestines of humans and animals is where it's commonly found. The cysts are shed in the feces. The fecally contaminated water that has the cysts in it is what's infectious. Ingesting contaminated water or ingesting food that has been contaminated by contact with contaminated water are the most common ways that it's transmitted to humans. Infectious dose. So this is the minimum amount that you would have to be exposed to. In this case, like I said, the cyst is the infectious phase, and as little as 10 could cause this infection, right? So that's very small. Incubation periods from the time you ingest it to the time you're potentially feeling symptoms can be typically about a week. So it's not instantaneous. I guess that's somewhat good, except for when you're trying to figure out where you got it from. The scary thing with this is that most people, about 50%, are asymptomatic, never show symptoms. So, I mean, I don't even think that Anthony looked at his poo because he was feeling bad. I think because he was just curious, right? He may have been asymptomatic. Diarrhea, if severe, can lead to dehydration, right? So that's of concern. One of my students in my morning class, her cats had this. And she said, boy, they just don't even tell you how bad the gas smells. It smells really bad. And so greasy stools that tend to float, abdominal cramps, nausea, vomiting, headache, mild fever. With chronic infection, it can even lead to weight loss, right, because of not absorbing your nutrients. So I've got a couple of vet techs in this class, so I definitely have a question for you guys coming up. So for acute illness, very fast onset, it's typically 1 to 2 weeks, but can be longer. Why is that? Why do you think the infection could be longer?
The immune system?
Your immune system is one thing. Right? You could get reinfected too. Right, if you're not sure where it's coming from. Chronic illness, milder illness, and slower, months or years with relapse even. And again, asymptomatic illness is one of the major reasons for its spread, right? They never feel sick. If you're asymptomatic, you don't know you're sick. You don't know you're harboring this, that you're spreading this infection. Who's at risk? Obviously immunocompromised or chronically ill, and then of course the very young, infants and children, right? Don't understand what's going on. People in daycare centers, those who come in close proximity with those who is already infected, and so they go into really specifics later, right? People who have unprotected anal sex, travelers to countries where Giardia is common, someone who drinks contaminated or untreated water, so camping, hiking, swimming, right? People who have it says people who have contact with infected animals, this risk is small because the type of Giardia that infects humans is often different from the type that infects animals. I don't think that's so. I don't think so.
We tell clients, like, Giardia is in nearly every rain puddle
Yeah.
in New Orleans. It's not something you just,
Yeah.
because people are thinking about, I would only get it if I went camping, so my
Right.
dog was only going to get it in the woods.
Right.
And it's like we see Giardia every day.
And if the clients aren't careful, they very easily could get it from their pets, I would think. I mean, because how is it getting into those pools to begin with? It's from the animals, right? Although people too, right? But that's the thing is with hiking and such, you know, like the cleanest water is not, like if it looks crystal clear, don't drink it. Right? It does not mean it's clean, right? It could be the most contaminated water you've ever experienced. So if you're not hiking in your own water, bring the proper filtration devices with you, right? You do not want to suffer later after your trip. So, yeah, puddles, like she said, in New Orleans and stuff, right? We've got so many drainage ditches here. And I think that was the issue with this particular case study. So, cysts, right, are really hard to detect in the stool. So one of the more effective ways of determining infection is they're going to do a fecal assay still, but in this case they're going to do a direct ELISA looking for the antigen. And in order to determine subspecies, though, they'd have to do molecular testing, right? So Flagyl is the brand name. Metronidazole is the drug name that's typically given. This can be given for bacteria as well as parasites. And the reason for is how this actual drug works. It it actually enters into the cells and interferes with their metabolic ability. And one of the things it does is it produces free radicals. These are really damaging forms of molecules. So I've been told that this is typically given for anaerobic bacteria, again, because of that particular type of pathway that it interferes with. So it's interfering with their metabolism. So how did this microbe get into the irrigation water supply? It's usually found in water polluted with fecal or manure contaminants. Activity of wild animals or livestock near a water source is the most common mode of transportation. So that may have been the case for this one. Remember the life cycle, you're going to ingest the cysts, those are going to come out, and 2 of them come out. The 2 trophocytes come out of the cyst. They can feed, this is impressions that they left from those sucking discs, from sucking on the mucus of your intestines. So the bad news with this one is that relatively resistant to chlorination and UV exposure and even freezing, these cysts. And actually our stomach pH is what activates, the low pH is what activates them to come out of the cyst form when they get to our small intestines. So, the most times we talk about how our stomach is protecting us because of those acids and will kill a lot of the things that we ingest. Not so in this case, right? Actually attributes to its ability to be infectious. So I had given you guys a copy of a report, right? So you'll see that in 2010, in July, August, September, and October, there was increases in the number of Giardia. What would you think this would be largely contributed to? Swimming in natural waters, right? Very common thing for those months throughout the year, especially July and August. So it goes through all the things that people in agriculture have to keep in mind to limit contamination. So I'm not going to bore you guys with that list and I'm way out of time. But as the theme has been for most of our case studies, right? Proper hygiene and hand washing are very important. Thank you guys for your patience.